Good morning everyone. How are students? Everything going well? Today rainy day is there. Raining is there in Vijayawada. I think at your places also raining is there. Are you enjoying today's weather? Good morning, good morning. I asked, are you enjoying? So some students are saying yes sir, some are saying no sir. Okay. Okay, great. Normal students enjoy this rainy day, right? And I don't think that no one had rain, right? Okay, some are saying that only cloudy weather, no rain. Okay, but here in Vijayawada, it is in night it is started, and now also raining is okay. So today we will understand work done by the ideal gas, how to calculate work done. Okay, so first, in ninth class you studied about work done. Can you tell work done formula? How to calculate work done? Do you know formula of this work done? What is the formula of work done? How to calculate work done? Any idea you have? Send your response. Okay, yes, very good, very good. Most of the students are sending F into S. Okay, very good, very good. F is force and S is displacement. Yes, you all know it. Very good. So if normally anyone asks you work done, then you will say it is force into displacement. You can use D, you can use S, whatever. But we can say that work done formula is force into displacement, and some students are saying that sir, it is F S cos theta. F is force, S is displacement, and cos theta also. Yes, actual formula is Fs cos theta, but normally we say that work done is force into displacement. But in reality, actual formula is Fs cos theta. Okay, so we will not go so much in this way mechanics. Just normally, if anyone asks what is the formula of work done, so you will say force into displacement. Why? Because we apply force on any object, then maybe that object start to move. So whatever that displacement, it is due to the force. More force, so maybe more displacement. That's why work done is force into displacement. You all agree? Yes. Now, here our topic is work done by the gas. Yesterday we discussed that for ideal gas we defined three quantities: pressure, volume, temperature, right? So here we do not talk about force, here we talk about pressure, because we discuss that this pressure is also arises due to force, because pressure equal to force by area. But here we talk about pressure, not about the force, right? Okay, so important point is what? For ideal gas, which formula we have to use to understand this point? First we have to understand the sense of work done by the ideal gas. This is the container. Okay, now in this container, this is, we are using a piston here, na? This is a piston. Okay, this is a piston and it is movable piston. This is what? Movable piston. Okay, now in this piston, suppose this ideal gas is filled. Okay, this ideal gas is a filled. Okay, ideal gas molecules are there. All ideal gas molecules move randomly. They move in all direction. Can you say here oxygen molecule is at rest? No, that oxygen molecule is also moving, right, in all direction. Okay, so these ideal gas molecules are moving inside the container. Okay, now what we are doing, we are heating it. We are heating this ideal gas. Okay, so this is a heater. By using this heater, we are heating it. Okay, we are heating it. If we are heating the ideal gas, then what will happen? You will say, sir, temperature will increase. Agree or not? Temperature will increase, so their speed also increases, because speed directly depend on temperature. Or we can say these molecules will move with higher speed because we are increasing the temperature. Agree or not? Okay. Now, if speed will increase, so these molecule will collide with, these gas molecule will collide with high speed with this piston. If gas molecule will collide with high speed with this piston, so what is the chance that this piston can move up? Right or not? This piston can move up, means there is a displacement. Right or not? If there is a displacement, means here work done is there. Agree or not? So if we are heating this ideal gas, then gas molecule energy will increase, their temperature will increase. Okay, so they will collide with higher speed. All gas molecule collide with wall of this container and this piston. If they collide with higher speed, so there is a chance that this piston can move up, means displacement. And if displacement, to work done. And you know work done formula, force into displacement. So force we can write, I already told you that here everything is pressure. So pressure is equal to force by area, then force is equal to pressure into area. What is A? A is area of this piston. Here all molecules are colliding, na? Molecules are colliding like this. So A is the area of this piston. Right? Now one more thing you have to tell. What is the volume of, can you tell volume of suppose this distance is x and this area A is area of cross section. Okay, can you tell volume of this particular part? How much volume of this part? Can you tell volume? Send your response. How much volume of this part? If A is the area and x is this distance, then volume is how much? Very good. All students are sending A into x. Very good. So volume is A into x. Okay. So here, before heating, suppose this distance is how much? L. Okay. So volume and this area is A. A is area of cross section. So here we can take A is what? Area of cross section. L initial L is initial height or initial length of cylinder. Okay. initial length, right? So how much volume? The volume of gas V is volume of gas, that is how much? So we can say that this volume is A into L. Okay. Now we are hitting it, so this piston will move up. Suppose this piston move by x distance. This piston move up by x distance. So this piston move up by x distance and this structure become like this. Like this. Okay. Earlier this piston was here at this level. Okay. So this distance was L and displacement is x. Okay. So how much work done? So you know work done formula, force into displacement. So how much displacement? X. So we can say that work done is force into displacement. Now force, how much force? Pressure into area. Here we can write first force into displacement. Okay. Now understand this point. You will study in work energy power chapter about work done. What? That work done formula F into D, it's okay, but this work done formula we can use only for a force which is constant. Right? Can you tell this force is constant always? No. Why no? Yesterday we discussed. What we discussed? That if volume changes, then 100% pressure changes. Right or not? If temperature remain constant. But normally if we think, then we can say what? We can say that here if we change the volume, then pressure slightly changes. So if pressure changes, the force is not constant. If force is not constant, then by which force we have to multiply with displacement, because force is variable. So in such cases, in integration you studied that if a quantity is variable, then we have to take help of integration in variable system. Okay, so here it is variable system. So we don't use F into D, we will use here which formula? Integration of F into displacement, but there we use small displacement. There we use small displacement. F dx. What is dx? Small displacement. Is it clear? Okay. Otherwise, in differentiation also you studied, we can calculate it like this. F is pressure into area. Okay. And D is what? Displacement. How much displacement here? X. Okay. So it become PAX. Can you tell me what is AX? What is AX? Can you tell what is AX? Send your response. Send your response. What is AX? Volume. All students are saying volume, but tell me which volume it is. AL also volume, na? What is AX? All students are saying volume. Yes, okay, I agree, volume, but AL is also volume, then AX is what? AX is what? Change in volume. What is AX? change in volume, because after heating this piston move up. How much it move up? This much. So this area multiplied by x, Ax is this volume, right or not? Ax is this part volume. So that much volume changes. So what is Ax? Ax is change in volume. So Ax is change in volume. And if change is there, then we can write this change by delta sign. So we can use here delta V. Is it clear? So Ax is delta V. So at the place of Ax, what we can write? We can write P into delta V. Agree? Okay. Now understand. If here we say that pressure is variable. If pressure is variable, then we have to take a small volume, and small volume we represent by dV, not by delta V. At the place of dx, if dx means small displacement, and if displacement is small, then volume change also small. So we will use here dV. If we will use dV, then we have to use integration. So this formula will become integration of P dV. Is it clear? This formula become integration of P dV. What is dV? dV is small change in volume. Okay, one more point. What? This integration this integration also equal to area of the pressure volume graph. So very important. This is also equal to area of the pressure volume graph. Is it clear? So what is work done formula? To work done formula F into D, okay, but this formula is used in mechanics. Here in case of ideal gases, formula of work done is integration of P dV, okay, and this is also equal to area of pressure volume graph, okay. P is here pressure, dV is small change in volume, okay. What is P here? P is pressure applied by ideal gas. Pressure applied by ideal gas. Clear? Okay. So we understood it. Yes, integration. Yes, one student is saying that integration gives area of the graph. Yes, correct. Integration means summation. Summation of what? Summation of small area, right? Small part area. So area of the pressure volume graph will give work done. So by two formulas by two method we can calculate work done. Integration of P dV, first, second, area of the pressure volume graph. So if in the question they give graph, then think about area. And if in the question they did not give this graph, then this formula we have to use. Clear to all of you? Okay. So our topic heading is calculation of work done by gas. So formula integration of P dV, and this is also equal to area of pressure volume graph. Now we will solve one question. Okay. Are you ready to solve question? So I think all of you are excited to solve question. This question. So read this question. Is it visible to all? This question you have to solve. What is given here? Pressure of 1 mole, so N equal to 1. Number of moles 1. Monoatomic gas increases linearly from, so this value is initial pressure. This is initial pressure to this value. This is what? Final pressure. What is the SI unit of pressure? Do you know SI unit of pressure is force by area. Force unit Newton, area unit meter square. So SI unit of pressure is Newton per meter square. Okay. Its volume increases from 0.2, this is what initial volume, to 0.5, this is what final volume. Okay, calculate work done by the gas. Our topic only work done. We will calculate only work done. If we know calculation of work done, then we can solve other also. So can we calculate here work done? So what I told, if graph is given, then we have to think about area. So area of force, area of pressure, volume graph will give work done. Okay. So this graph is given like this. Okay, straight line like this. How to find area? To understand, what you have to do, join the initial and final point with the volume axis. Listen each and every line. Join the initial and final point of the graph with the volume axis, not with pressure axis. So this is pressure axis, this is volume axis. This is initial point, this is final point. Join with volume axis. So this is volume axis, joined. Okay. This is volume axis, joined. Okay. Now what is it? What is it? It is trapezium. Agree or not? It is what? Trapezium. If it is trapezium, so what is the area of trapezium? What is the area of trapezium? So area is equal to, do you know area of trapezium? Can you calculate area of trapezium? This point is given 0.2, this point is given 0.5, this point is given 4 into 10 to the power 5, and this point is given 8 into 10 to the power 5. Can you calculate this area? I am asking you this area. This is trapezium. Do you know formula of area of trapezium? Yes, very good. Some students are sending correct. Area of trapezium is sum of parallel side multiplied by distance between them divide by 2. This is the formula of area. Yes, very good. Most of the students are sending correct. Okay. Now put the value and find it. So which are parallel side? This side and this side. This side and this side. These are parallel side. Agree or not? Now this side is how much? This side is how much? This side is 8 into 10 to the power 5. This side value is how much? 8 into 10 to the power 5. Okay. And this side value how much? 4 into 10 to the power 5. So sum of parallel side, one side is 4 into 10 to the power 5, another side 8 into 10 to the power 5. Okay. Multiplied by distance between them. This is distance between them, right or not? This much. This is the distance between them. So this total value is how much? This total value is how much? This is 0.5. This total value 0.2. So this value is 0.3, right? So this value is 0.3, right? Okay. So multiply it with 0.3, right? Multiplied by 0.3, divide by 2. So 4 plus 8, 12. So it become 0.3 into 12, 10 to the power 5, we can take common, divide by 2. So 12 by 2, 6 into 3, 18, 10 to the power 5. So 1.8 into 10 to the power 5. This is our answer. Okay. So 1.8 into 10 to the power 5 joule. This is our answer. Clear? Understood? So by graph we calculate work done here. Now next question. You all agree? Understood? Okay, very good, very good. Now next question. Before we will solve this question, okay. Now in this question, you have to calculate this work done. 1 mole of ideal monatomic gas is taken round the cycle ABCA. This cycle is given. This volume is V0. This volume 2V0. Pressure P0. This pressure 3P0. So first question I am asking you. Tell me the work done by the gas. To solve and tell me the answer. How much work done by the gas? Send your response. I am waiting. How much work done by the gas? Other part also we will solve, but first one by one. First part work done we understood. Then next part we will discuss about heat and internal energy. That also we will discuss. Some students are saying 0. Work done is 0. No. Work done is not 0. Yes, this what is her name? Reddy Subhasini. Very good. Correct. You send correct answer. Your answer is correct. Reddy Subhasini. Okay. Most of the students are saying 0, 0, 0. That is wrong. That is wrong. Okay. Viber. This student earlier also he given correct answer. Viber correct. Very good. Okay, to understand how to find work done. Very simple. What I said. Work done is equal to area. Work done is equal to area. So here it is closed loop. It is closed loop. And if closed loop is there, then area of the loop is equal to work done. So here you can write this definition also, that work done is equal to area of pressure volume loop. Pressure volume loop. So if in the question they give loop, okay, see here, this is not a loop. Process start from A to B. This is not a loop. It completed at B. It is not closed cycle. Right or not? It is not closed cycle. So in such cases, what you have to do, join the starting point and final point with volume axis, join them to be joined. Okay, then we calculated area of this loop. If in the question directly they give loop, so here in this question directly loop is given. Now no need to join with volume axis because already it is a loop. So in such questions, work done is equal to area of this loop. So this complete loop area we have to take. Okay, and here also I mentioned that area of pressure volume graph. So this is for, if closed loop is not given, then you have to make closed loop. How? By joining the initial and final point with volume axis. And if loop is given, then directly area of pressure volume loop, closed cycle. Closed cycle. Clear? Okay. Now next. So in this question, we have to find out the work done. Now you will say, sir, it is not 0 because area of this loop is not 0, right? It is a loop, 100% area is non-zero. So how much area here? So now easily you can calculate. You all can see this is what triangle, and right angle triangle area, you all know area of triangle is area of right angle triangle is 1/2 base into height. So this is what, this is base. So we need this much distance. This is what, this is perpendicular or height. So we need this distance. Agree or not? This total 3p0, this much, this total 3p0, this much p0. So 3p0 minus p0. So this distance is 2p0. Okay. This distance 2v0, this distance v0, then this will be 2v0 minus v0. So it is how much? v0. Right or not? So this distance, this distance is how much? v0, and this distance is how much? 2p0. So 1/2 into base is v0, and height is 2p0. So 2 cancel out, so it is how much? p0 v0. This is the area of the right angle triangle. Is it clear? So our answer is work done by the gas is equal to p0 v0. Okay, understood? So it is clear. Now we can easily calculate work done. Okay students, now understand other parameters. In ideal gas, work done is most important. Then next is heat and internal energy. You know how I started work done story? First, what we did? Here we supply heat, right or not? We supply heat. Due to supplying heat, these gas molecule temperature increases. So they will move fastly. They will collide with high speed. Due to that, piston started to move up, right? So how this work done arises? What is the cause of work done? Heat supplied in this particular example, right or not? Means heat supplied and work done both are related. Agree or not? Yes. So whatever heat we supply, this heat supplied can be utilized in two parts. First part, all gas molecule, their temperature can increase. Okay, if their temperature increase, then what will happen? Their internal energy will increase, right or not? Because gas molecule internal energy means that is due to the temperature, right or not? So here we have to define. You say, sir, we did not hear till now about internal energy. We heard about potential energy and kinetic energy. Yes, correct. Correct. Internal energy is nothing. It is the sum of kinetic energy and potential energy. So gas molecules are moving means kinetic energy, and potential energy is due to the orientation. You studied potential energy? Yes. But here in case of ideal gas, we assume that potential energy is 0 for ideal gas. So we only talk about kinetic energy. So total internal energy is equal to kinetic energy because potential energy is 0. We assume it. Okay. So internal energy is equal to kinetic energy. So we will understand about internal energy and this heat supplied in next class. Then we relate work done, heat supplied and internal energy in tomorrow class. Is it clear? Okay, so today in this class we understood about how to find work done. Okay, and we solved some examples also. In tomorrow class we will relate heat and work done and internal energy. First we will understand about internal energy, then we relate them. Okay friends? Okay. So I hope you understood this work done calculation, and thank you for listening the class. Further we'll discuss tomorrow. Okay friends.
Good morning, students. So today we are going to start oxidation number calculations. In last class we have discussed about rules that how to find out oxidation number. So let us start with today's questions. So today I will give you some questions. You have to tell me oxidation number of underlined species. So question is calculate oxidation number of underlying atoms in following. First I am writing all the questions, then I will write here underline that which you have to find out. K2MnO4 Na2CrO4 HNO3 H2S2O3. So for which species we have to find out? In first manganese, second manganese, chromium, nitrogen, sulfur. So try to solve this. So today we'll only practice questions based on this oxidation number. So you'll get a lot of questions. Start solving these. You have to tell all 6 parts in single message. Do not write first, second, third, fourth. If we have to multiply it by charge, no sir, which is called oxidation state or oxidation number. Why we have to multiply by charge? We are not multiplying by charge. Sum of oxidation number of all atoms. In neutral molecules, sum of oxidation number is equal to 0. Start solving all these. Today we'll get nomenclature exercise. Today in Telegram you will get nomenclature. Solve this, you will get 2 minutes. Then after that I will solve all these. Here we are not talking about oxidation state. We are talking about only oxidation number. In next class we'll discuss oxidation state. In this class only write oxidation number. Plus 7, 6, 5, 6, 6, 2. Seven, 3, 6, then 5 and 2. Summary plus 7 plus 6, 6, 6, 5. Correct. Maximum students are getting correct answer. Very good. So let us start solving. How to find out? In first manganese. First potassium. Potassium is alkali metal and we have discussed that for alkali metal oxidation number is +1. So for potassium +1. For manganese we have to find out. So what we take X. How many oxygen atoms are present? 4. And oxygen shows which oxidation number generally? -2. So 4 into -2 is equal to 0. So what we get? Value of X is +7. -8 here +1. So -7. So X here will go and what is formed? +7. So in first one oxidation number of manganese is +7. Okay. Now if we solve second, again what is given? Potassium, but how many potassium are given? 2. So 2 into plus 1, 1 potassium and 2 potassium are present. Now next, for manganese, X, 4 into minus 2 is equal to 0. So it is minus 8 plus 2, minus 6. X minus 6 is equal to 0. So what we get? Value of X is plus 6. So oxidation number here for manganese is plus 6. First compound KMnO4 is called as potassium permanganate, and this K2MnO4 is called as potassium magnate. So plus 7, and next is plus 6. Now let us move to Na2CrO4. How many sodium are present? 2. And sodium is alkali metal, and for alkali metal what is oxidation number? Plus 1. Now for chromium, what we take? X. Now 4 into minus 2 is equal to 0. So what we get here? Minus 8, here plus 2. So what is present here? Left plus 6. So oxidation number of chromium here is plus 6. Clear? Let us move to next one. K2Cr2O7, 2 potassium. So 2 into plus 1. Now how many chromium are given? 2. So 2 into charge on 1 chromium is X. Let us say oxidation number is X. Plus 7 into minus 2 is equal to 0. So 14, 2. What is left? 12. So 2x is equal to 12. So value of X is plus of 6. So here oxidation number of chromium is plus 6. Now this Na2CrO4 is called as sodium chromate, and K2Cr2O7 is called as potassium dichromate. Let us move to next, HNO3, nitric acid, hydrogen plus 1. Now nitrogen we have to find out. Let us take X. 3 into minus 2 is equal to 0. So what we get here? X is equal to plus 5. So what is oxidation number of nitrogen here? Plus 5. Nitric acid. Now next, how many hydrogen are present? 2. And hydrogen generally shows which oxidation number? Plus 1. So 2 into plus 1. Next, how many sulfur are present? 2. So 2 into X. Oxygen 3 into minus 2 is equal to 0. So minus 6 plus 2. So 2x is equal to 4. So value of X will be plus 2. So here oxidation number is plus 2. And this H2S2O3 is called as thiosulfuric acid. What is name for this? Thiosulfuric acid. So questions can be asked like this also, that what is oxidation number of sulfur in thiosulfuric acid? So what is oxidation number here? Plus 2. What is oxidation number of nitrogen in nitric acid? What is oxidation number? Plus 5. Just check, any doubt in these 6 examples? Just check, any doubt in these 6 examples? Then we will move to next other examples. Clear? Okay. Now write down next 7th is HClO4, iO3 negative, ClO2 negative, ClO negative, NO2 negative, SO3 2 negative. Now for which species or underlined species we have to find out here? Chlorine, iodine, chlorine, chlorine, nitrogen, sulfur. Now what is given here? Charged species are given. So in this 8, 9, 10, 11, 12, sum of oxidation number is equal to charge present on that polyatomic ion. Solve these parts. After 2 minutes we'll discuss all these. You have to send all answers in one message. Do not write separately, otherwise I will not able to read. Some plus 5, 7, 5, 3, 1, 3, 4, 7. Next we are telling 5, 3, 1, 3 and 4. Very good. Many students are writing correct answer. 8 is nowhere. Some Naved is writing plus 8. There is no oxidation state here plus 8. Maximum students are writing correct. Very good. 7, 5, 3, 1, 3, 4. Positive oxidation state. Very good. Let us start solving these. Now what is given? Hydrogen. For hydrogen, plus 1. Now for chlorine, what we take? X. 4 into minus 2 is equal to 0. So what we get here? Minus 8 plus 7. Plus 1 means plus 7. So in HClO4, what is oxidation number of chlorine? Plus 7. Now let us move to second part. What is given? Now charge species. So iodine, but we take X. How many oxygen? 3. And oxidation number of oxygen is minus 2. Now sum is equal to charge. What is charge here? Minus 1. So what we get here? Plus 5. So oxidation number of iodine in this compound is plus 5. Now next is ClO2 negative. So X, how many chlorine? 1, 2 oxygen, 2 into minus 2 is equal to minus 1. So minus 4, where it will go plus 4, minus 1, what do you get? Plus 3. So here what is? Plus 3. Now here chlorine, X, 1 oxygen only, minus 2 is equal to minus 1. So what we get here? Plus 1. So chlorine will show here plus 1. Now if you remember, we have written rule for halogen. Halogen generally shows minus 1, but fluorine always minus 1. Chlorine, bromine, iodine can show minus 1 to plus 7. So if you check, chlorine is present in plus 7, iodine present in plus 5, chlorine present in plus 3, chlorine present in plus 1. Because here chlorine is combining with more electronegative element, oxygen. That's why chlorine is showing which oxidation state here? Positive oxidation state. Okay. Now next, NO2 negative. Now nitrogen, X, how many oxygen? 2. So 2 into minus 2 is equal to minus 1. So what we get here? Plus 3. So NO2 negative is called as nitrite ion. So in nitrite ion, what is oxidation number of nitrogen here? Plus 3. Now next is sulfur. Let us take X. 3 into minus 2 is equal to minus 2. So what we get here? X is equal to plus 4. X is plus 4 means oxidation number of sulfur is plus 4. Now this SO3 2 negative is called as which ion? Sulfite ion. Remember, it is not sulfide, ITE, sulfite ion. NO2 negative is called as which ion? Nitrite ion, ITE. So NO2 negative, nitrite, SO3 2 negative, sulfide. Any doubt in these 7 to 12? Is oxidation number also a charge? It is charge. When all bonds are broken, go to last class where we have written definitions. What is oxidation number? Listen those earlier classes. Now let us move to next part. Now next questions are H3PO4 phosphoric acid, H3PO3, H3PO2, N2H4, H4P2O7, C2O4 2 negative. Now for which? Phosphorus, 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 nitrogen, phosphorus, carbon. Solve all these. So we are calculating average charge. If more than one atom is present, then it is average charge. In H3PO2 only one phosphorus. So what is average? Average and it is same. If similar type of more than one atoms are present in different oxidation state, then average will be different. Unnecessary, you are confusing yourself. Solve only these questions. Five, 3, 1, 2, 5 and 6. Carbon, how 6 is possible? Carbon cannot show plus 6. Maximum for carbon is plus 4. Carbon range is -4 to +4. It cannot show plus 6. Five, 3, 1, -2 plus 10 by 3. Where 10 by 3 is coming? 10 by 3 is now where 2 phosphorus are there, not 3 phosphorus. Plus 5, plus 3, plus 1. Next -2 you are saying. Next is plus 5, plus 3. It seems to be correct. Plus 3, plus 5, plus 3, plus 1, -2, plus 5, plus 3. This seems to be correct. Let us start solving. First, how many hydrogen are present? 3. So hydrogen shows +1 oxidation number. Now only one phosphorus. So what we take? X. 4 into -2 is equal to 0. So what we get here? X is equal to plus 5. -8 plus 3. So -5. So X is equal to plus 5. So here phosphorus is present in plus 5. H3PO4 is called as phosphoric acid. What is the name of H3PO4? Phosphoric acid. Now next H3PO3. It is called as phosphorus acid. Name for H3PO3 is phosphorus acid. So 3 hydrogen are present. 3 into plus 1 phosphorus X. 3 into -2 is equal to 0. So now what you get here? -6 plus 3. So what we get here? X is equal to plus 3. So in phosphorus acid, oxidation number of phosphorus is plus 3. And in phosphoric acid, plus 5. Now next is H3PO2. H3PO2 is called as hypophosphorus acid. What is name? Hypophosphorus acid. Now 3 hydrogen are present. So 3 into plus 1 for phosphorus X. 2 into -2 is equal to 0. So -4 plus 3. So what we get? X is equal to plus 1. So in hypophosphorus acid, phosphorus is present in which oxidation number? Plus 1. Now next N2H4. N2H4 is called as hydrazine. What is name? Hydrazine. Now 2 nitrogen are present. So what we take? 2 into X. 4 hydrogen. Hydrogen shows plus 1 is equal to 0. So what we get here? X is equal to -2. So here in hydrazine, nitrogen is present in which oxidation number? -2. Now next is H4P2O7. This is called as pyrophosphoric acid. What is name? pyrophosphoric acid. So how many hydrogen are present? 4. So 4 into plus 1. How many phosphorus are present? 2 phosphorus. So 2 into X plus 7 into minus 2 is equal to 0. So 14, 4, 10. So 2X is equal to plus 10. So what is value of X? Plus 5. So in H4P2O7, phosphorus is present in which oxidation number? Plus 5. Okay. Now next C2O4 2 negative. This is called as oxalate ion. C2O4 2 negative is called as which ion? Oxalate ion. Now 2 carbon are present. So what we take? 2 times of X, 4 oxygen, 4 into minus 2 is equal to charge present. What is charge here? Minus 2. So if we solve here, what we get? X is equal to, here it is 4 minus 6. So it will become plus 6. So plus 3. In oxalate, carbon is in which oxidation number? Plus 3. Any doubt in these? You should remember name of these compounds also. Hydrazine, oxalate, H4P2O7 is pyrophosphoric acid. Today we'll do lot of question. Do not take tension. Give more examples. Any doubt in these example up to 18? Okay, let us move to next one. Write down next. Arsenic sulfide, As2S3. S5 also. Carbonate ion CO3 2 negative. Bicarbonate HCO3 negative. Bioxalate ion HC2O4 negative. Next is S4O6 2 negative. For here arsenic, carbon, sulfur. Solve these all. Plus 3, 5, 4, 4, 3, 5 by 2. First, what you are saying? Plus 3, second, plus 5, 4, carbonate, bicarbonate, 4, correct. Next, 3 and 5 by 2. Correct. These are correct answers. What are correct? Plus 3. Next is plus 5, plus 4, plus 4, plus 3 and this will become 5 by 2 plus 5 by 2. Let us start from first one. Now this is simple compound. If you remember in class 9th we have studied about this AXBY. If you break this, first cation A plus Y and this B is anion X negative. And this cross method you have studied for writing formula. So this same you can apply here. So what we write here, arsenic plus 3, sulfur is present in -2. And so directly we can write arsenic shows 2 oxidation state, plus 3 as well as plus 5. So simple you can cross multiply, plus 3 -2. Here sulfur -2 is sulfide ion. IDE, sulfide ion. It is different, it is different. And similar we can write here cross multiply, plus 5, here it is -2. So in first one oxidation number of arsenic is plus 3, in second one oxidation number of arsenic is plus 5. So simple cross method we can apply here. Now in case of CO3 2 negative, CO3 2 negative is called as which ion? Carbonate ion. Now carbon if we take X, so 3 into -2 is equal to -2. Sum of oxidation number is equal to charge present. So what we get here, -6 -2. So carbon is present in which oxidation number? +4. There. Let us move to next part. HCO3 negative is called as which ion? Bicarbonate ion. So hydrogen if we take +1, 1 carbon X, 3 into -2 is equal to -1. So what we get here? -6 and here it is +1, so -5. So it will become +4. So in bicarbonate also carbon is present in which oxidation number? +4. So carbonate, bicarbonate same, carbon is in +4 oxidation number. Next HC2O4 negative, this is called as bioxalate ion. Whenever one hydrogen is left, what is used? Bi is used. Now how many hydrogen are present? 1. So +1. How many carbon are present? 2. 2 into X, 4 into -2 is equal to -1. So what we get here? 2x is equal to 2x is equal +6. Value of x is equal to +3. So in bioxalate, carbon is present in which oxidation number? +3. Now next S4O6 2 negative. This is called as tetrathionate ion. What is name for this? Tetrathionate ion. Now 4 sulfur are present. So what we take? 4 into X. How many oxygen are present? 6. 6 into -2 is equal to total charge -2. So 4x is equal to 10. So value of x is +5 by 2. So oxidation number of sulfur here is +5 by 2. That is 2.5. Now check, any doubt in this up to 24? All are correct. Maximum students have got this correct answers. Clear? Now let us move to 3 more examples on this. Okay. Next is 25th Fe3O4. 26th is Pb3O4. 27 is CaOCl2. So only have to tell me oxidation number of iron, oxidation number of lead, oxidation number of chlorine. Oxidation number, not any oxidation state. Solve these. These are easy, only oxidation number you have to tell. 7 by 3 is not answer. 8 by 3, 8 by 3, 0. Piyush, it is correct. And 4 you are writing. 4 is not correct. 8 by 3, 8 by 3, and for last it will be 0. Not 1 by 2. How you are getting 1 by 2? Calcium is alkaline earth metal. Plus 8 by 3, plus 8 by 3 and 0. That is looking correct. Plus 1 by 2 is not possible for chlorine. Now these are some special cases. So first we find oxidation number. How many iron are present? 3. So 3 into x. 4 into minus 2 is equal to 0. So what we get here, x is equal to plus 8 by 3. This is oxidation number of iron. But if I write some points about this, that Fe3O4 is a mixed oxide. Now what is meaning of mixed oxide? So first we have to listen about what is oxide. Binary compound of oxygen. Binary compound means where two elements are present, one oxygen and one other element, that is called as oxide. Now oxide are of two types, one is simple oxide and one is mixed oxide. So if any metal or element is present in only one oxidation state, that is called as simple oxide. If element is present in more than one oxidation state, that is called as mixed oxide. So in case of this Fe3O4, it is a mixed oxide of ferrous as well as ferric. So this Fe3O4 exists as Fe3O4 exists as FeO dot Fe2O3. FeO is ferrous oxide, Fe2O3 is ferric oxide. Now in ferrous oxide, iron is in plus 2 and in ferric oxide, iron is in plus 3. So if they ask question that what is oxidation state of iron, not number, what I am writing, oxidation state. Now in FeO only one iron is present. What is oxidation state? Plus 2. In case of Fe2O3, how many iron are present? 2. And what is oxidation number? Plus 3 means state. Plus 3 and plus 3. So if they ask what is oxidation state of iron, oxidation state is plus 2, plus 3 and plus 3. One iron is in plus 2 oxidation state, 2 iron are present in plus 3. So this we can only find if we remember this, that Fe3O4 is FeO dot Fe2O3. Now if we write oxidation number, average of all these, so plus 2, plus 2, plus 3 divided by 3. So what you get here? Plus 8 by 3. Same we are getting here, plus 8 by 3. So what we have to remember, that oxidation state is plus 2, plus 3, plus 3. And average of these oxidation state is oxidation number. What is oxidation number? Plus 8 by 3. Now let us move to Pb3O4. This is also a mixed oxide. So first if we find 3 times of x, 4 into minus 2 is equal to 0. So what we get? X is equal to same plus 8 by 3. This is oxidation number of lead, not oxidation state. So this Pb3O4 is also a mixed oxide. Is mixed oxide. Clear? So if I write how this will exist, Pb3O4 exists as 2 times of PbO dot PbO2. Lead monoxide and lead dioxide. This PbO is lead monoxide, PbO2 is lead dioxide. So in PbO, what is oxidation number of lead? Plus 2. And in PbO2, what is oxidation number of lead? Plus 4. So if they ask what is oxidation state of lead in Pb3O4, now if we check 2 times of PbO, means 2 lead are present in plus 2. So plus 2, plus 2. And 1 lead is present in plus 4. So what is oxidation state of lead here? Plus 2 and plus 4. And if I find oxidation number, average of this, so sum of all these divided by total number of atom 3. So what we are getting? Plus 8 by 3. Same what we are getting, plus 8 by 3. So if they ask in Pb3O4 what is oxidation number, 8/3. What is oxidation state of lead? 2 lead are present in +2 and 1 lead is present in +4. So this we can only find out if you remember these are mixed oxide and how they exist. Okay. Now next is CaOCl2. This you have heard. This is called as bleaching powder. One component of bleaching powder is CaOCl2, class 10th, acid, base and salt. This compound bleaching powder. In 10th class you have written calcium oxychloride, but when we move in class 11th we write this as different name, not calcium oxychloride. So if I write here, how many calcium are present? 1 plus 2. How many oxygen? Minus to 1. 2 times of X is equal to 0. So what is oxidation number? X comes out to be 0. Now this is oxidation number of chlorine, that is average of the oxidation state. Now how this CaOCl2 exists? This CaOCl2 exists as CaOCl and Cl. Now which type of ions are present here? If I break this, it contains calcium +2, ClO negative and Cl negative. What is present here? ClO negative, Cl negative. In ClO negative, if I find oxidation number of chlorine, so it comes out to be +1. And here what is oxidation number? -1. So here 2 chlorine are present. 1 chlorine is in +1, 1 chlorine is in -1. So if I write what is oxidation state of chlorine, so what answer you will write? +1 and -1. And if I find oxidation number average, so +1 -1 divided by 2, what we will get? 0. So in bleaching powder, 2 chlorine atoms are present, and in those 2 chlorine atoms, one is present in +1, one is present in -1. So if we find oxidation number, average will become 0. Here, any doubt in these 3 examples? These are important cases which we have to remember, these Fe3O4, Pb3O4 and bleaching powder. Check this, any doubt in these 3 examples up to 27? Okay, now last 3 very simple examples I am giving. Just tell, these are special examples. Now 3 simple examples. Last 3 examples: Na2O2, KO2, KO3. Now, last 3 examples, we have to tell oxidation number of oxygen in these 3 compounds. This CaOCl2 will be called as calcium hypochlorite chloride. OCl negative is hypochlorite ion, Cl negative is chloride ion. If you write complete IUPAC name, but you can call that as calcium oxychloride also, no problem in that. Now with how +2, 4, 6? You are saying oxygen is showing +6 oxidation state. How it is possible? Not possible, no? Oxygen only shows positive oxidation state with fluorine, not with this sodium potassium. If you take here, 2 sodium are present, 2 into +1, 2 into X is equal to 0. So what you get here? X value is -1. So this is called as peroxide. Sodium peroxide. In peroxide, oxidation number of oxygen is -1. Now in KO2, potassium +1, 2 oxygen, 2 into x is equal to 0. So what is value of x? Minus 1 by 2. This is called as superoxide. Potassium superoxide. And in last, again potassium plus 1. 3 into x is equal to 0. So what is value of x? Minus 1 by 3. This O3 negative is called as ozonide. Potassium ozonide. This is sodium peroxide. This is potassium superoxide. This is potassium ozonide. So these are questions based on oxidation number. Clear? So in next class, what we discuss, how to find oxidation state. For that, structures are required, and one new topic, that is charge transfer. Okay. So students, thank you for today. In next class, we start about oxidation state.
Hello, good morning, students. Good morning, all. Yeah, Anju, good morning. Okay. Lekya, yeah, good morning. I'm fine, Amma, I'm fine. I'm good. Yeah, very good morning. Okay. Good morning. Yeah, good morning, butterfly. Balu, yes. Good morning, all of you. Did you read yesterday's topic? Yeah, Lakshmi Pujita. Yes, good morning, ma'am. Yeah, I'm good, Sirisha. Yeah, Bhavesh, Banusha. Yes. I'm good. Okay. Did you read yesterday? AR. Okay. Sairaj, very good. Yes. Sanjana. Yeah, I'm good, Amma. Anju. Okay. That is so much. Okay. Okay. Fine. Quickly, let us have the questions, and after that, we'll proceed to the today's topic, that is fungi. Okay. So now, yesterday I have discussed about the Protista. So in that Protista, okay, I want the answers, okay, for some of the questions. So Protista include only unicellular eukaryotes or unicellular prokaryotes also. Unicellular eukaryotes only or unicellular prokaryotes also. Unicellular eukaryotes or unicellular prokaryotes or both of them. Unicellular eukaryotes only. Very good. Unicellular eukaryotes. Correct. There are no prokaryotes in the Protista. They are all unicellular eukaryotes. Very good. Okay. So now, let us proceed with the other things. Tell me, among the photosynthetic protists, I have given you like 3 groups for their photosynthetic protists, consumer decomposer protists and protozoan protists. Among these 3, which ones will be performing photosynthesis with the help of chlorophyll A? Among these ones, which ones will be performing photosynthesis, yes, with the help of chlorophyll A? Chlorophyll A. Euglena, very good Tikshana, euglena, very good, correct, euglena, very good Amma, too good. Lakshmi, cyanobacteria, no, euglena, cyanobacteria will come under Monera. Yeah, because euglena will have the same pigment like the plants. Yeah, cyanobacteria is also correct, but it is a moneron. I have asked the question about protists. So cyanobacteria and euglenoids, they are similar to green plants in terms of having chlorophyll A. Okay. Next. Among the protists, which one releases toxins into the water bodies? Toxins. Diatoms, dinoflagellates, euglenoids. Which ones? They release toxins. Diatoms, Balu, no. Sairaj, correct, dinoflagellates. Balu, diatoms is wrong. Yeah, it is dinoflagellates. Dinoflagellates. Okay, good. Not diatoms. Not diatoms. The dinoflagellates will be releasing the toxins into the water bodies. I said yesterday, orally, right? Yeah. Among these 3, diatoms, dinoflagellates and euglenoids, which ones will be looking like, the structure will be looking like a soap box? Soap box. Which one looks like soap box? Yeah, Sanjana, red tides, correct. That is over. Yes. Samin, yes. Diatoms, yes. Balu, yes. Diatoms. Soap box structure is there for diatoms, which will come under chrysophytes. Correct. Right. What is the name of the body of slime molds called as? Body of the slime molds, it is called as body of the slime molds. Slime molds body, it is called as. Very good, Sairaj. Very good. Plasmodium. Very good. Correct, correct, correct. Yes, Sanjana. Yes, Plasmodium. Very good. Yes. Good. Now, if it is a protozoan, if Plasmodium is a protozoan, then it causes a disease. What is that? Plasmodium, if it is a protozoan. Okay? If it is protozoan, it causes a disease. What is that? Yes, Sairaj, very good. Very good. Harika, very good, very good. Malaria, malaria, yes. Good. Malarian parasite. Right. Very good. Correct. Two more questions, last two questions. Name the protozoan protist with cilia. Name the protozoan protist with cilia. Euglena, Paramecium, Plasmodium, Trypanosoma. With cilia, I am asking the organism name, not ciliates. Yeah, ciliates they are called, group. Samrin, yes. Ciliates, yes, yes, very good. Harika, very good. Paramecium, yes. Paramecium. Name the organism with cilia. Okay, Paramecium. Correct. Next one. Okay, what is the similarity between Trypanosoma and Euglena? What is the similarity between Trypanosoma, Euglena? First option, both can perform photosynthesis. Both can perform photosynthesis. Yes, very good. Sairaj, yes. Flagella, both will have flagella. Okay, both of them are having the flagella. Good. Which disease is caused by the Trypanosoma? Trypanosoma? I said orally yesterday. Both has flagella. Yes, is correct. Both have. Very good. Kiran Kumar, sleeping sickness. Very good. Tikshana, very good. Sleeping sickness. Correct. African sleeping sickness, it is caused by the Trypanosoma. Correct. Sleeping sickness. So, I hope all of you have studied thoroughly. Right? Yes, African sleeping sickness. Very good. So, we have in R.H. Whittaker 5 kingdoms are there. In that 5 kingdoms, Monera is the first one. Second one is the Protista based on the evolution. And the third one is fungi. So, now let us see the fungi. The next topic, next kingdom, fungi. It is also called as Mycota. Mycota. Another name of the fungi kingdom is Mycota. And study of fungi is called as mycology. Mycology, study of fungi. And about this fungi, the mode of nutrition, okay, they don't have any chlorophyll. They are the heterotrophic organisms. They will show only heterotrophic mode of nutrition. Let us first see the general characters, okay, and the briefly explanation about the classes. So, the first one, all of you can see this colorful picture. These are toadstools which are poisonous fungi. So, if these white spots are there on the red ones, it is the poisonous fungus, and the name of this poisonous fungi are toadstools. So, these are non-edible. If you eat this kind of mushrooms, poisonous mushrooms, will be having certain disorders like indigestion. So many other problems will be coming. Okay, so these are the poisonous fungi. Toadstools are poisonous fungi. These are one among them. Fungi. This is mushroom, the edible one. Mushrooms. Mushroom scientific name is Agaricus. Agaricus bisporus. Agaricus is the genus name. So first name of the mushroom is, yes, that is the mushroom scientific name, Agaricus. These are edible. We can eat them. They are the rich source of proteins, vitamins and minerals. Proteins will be there, vitamins and minerals will be there. So we can have this mushrooms, edible one. But the previous one we are not supposed to eat because these are poisonous one. You can see here these are poisonous fungi. So toadstools are poisonous fungi, whereas mushrooms are edible fungi. Okay. Then next, these are another fungi. These are yeast. Yeast or unicellular fungi. This is the only one unicellular fungus in your syllabus. Unicellular edible fungus. Okay. So yeast is unicellular and it is edible fungus, and we can use this yeast in the bakery industries and in the brewing industries like that. The scientific name of yeast is Saccharomyces cerevisiae. Saccharomyces cerevisiae. The scientific names are important, so you have to learn the scientific names after coming here. Okay, so unicellular edible fungus is there, only one, that is the yeast, and it is used in bakeries and brewing industries, and its scientific name is Saccharomyces cerevisiae. This is only unicellular. Now coming to these molds. The molds are fungi which are filamentous forms. These are the filamentous ones. They all will be having the filaments. These are all hair-like things or filament-like things will be there in this. They are called as molds. These are another type of fungi. You can see here, this is penicillium. Penicillium, you must have known about penicillin antibiotic. Penicillin antibiotic. Alexander Fleming discovered that antibiotic which is used in the Second World War for the treatment of so many soldiers, wounded soldiers. That penicillin antibiotic is obtained from this penicillium fungus. So this is useful one because from this we are getting the penicillin. Penicillin antibiotic is obtained from the fungus penicillium, and it will be looking like this. Penicillium. Okay, this is the fungus and it will be giving us penicillin, the antibiotic, which is used to treat several bacterial infections. Okay, generally to treat the bacterial infections, this is useful, the antibiotic penicillin. And that penicillin will be acting on the cell walls of bacteria. I have told you in the Monera that cell wall of bacteria or Monera, it is made up of peptidoglycan. So it will be breaking that peptidoglycan or polysaccharides and amino acids. So to break the cell wall of bacteria, generally this penicillin is useful. The penicillin antibiotic which is obtained from the penicillium. Okay, this is useful fungus. Even yeast is also useful fungus. But it is not necessary that all the fungi are useful. There are some fungi which are pathogenic also. They will cause diseases in the plants, humans, animals, etc. So if you see the mustard plant, on the mustard plant, we will be seeing the white spots that are caused by one fungus. And some plants will be showing iron type of rust, that is called as rust disease. Okay, that is caused by one more fungus, that Puccinia. Smuts will be there, that is caused by Astralago. So some pathogenic fungi are also there, some are useful fungi. So both are there. They are useful, they are harmful also at the same time. Okay, but the one organism, one fungus cannot be harmful and cannot be useful at the same time. Some are useful, some are harmful. Okay, that is with respect to the fungi. These are all again filamentous forms only. Filamentous forms. You can see here, just I was talking about this rust only. this is a wheat plant. On that wheat plant you can see this iron rust, iron-like rust. We'll be seeing on the iron rods and all. Okay, if it is not painted properly, they'll be having the rust formation will be there without paint. So that kind of rust will be formed by the fungus Puccinia. It is called as Puccinia. So this is causing the plant disease. Rust disease is caused by the Puccinia. This is harmful one because it is causing the disease in the plant. The next one, coming to the fungi general characters. Fungi general characters. All those fungi, they are cosmopolitan in distribution. That means they are present everywhere. They are present in the air, soil, water. Okay, cosmopolitan. Cosmopolitan in distribution. Present almost everywhere. Then these are generally, they are eukaryotes. No doubt they are all eukaryotes. Except one, they are all multicellular eukaryotes. Multicellular filamentous eukaryotes. Except one. What is that except one? The exception is yeast. Yeast is the only one unicellular edible fungus. Remaining all are multicellular filamentous eukaryotes. Okay, eukaryotes is applied for all the fungi. So these are multicellular, made up of many cells, and they will be arranged in the form of a filament where one above the other all the cells will be there like this filament. This is called a filament. They will be present like this. Okay. Then these cells, they have cell wall. The outermost wall layer will be the cell wall. This cell wall is made up of chitin and polysaccharides. Chitin will be there, chitin or chitin, and even polysaccharides are also present. Chitin also is a polysaccharide basically, but there can be other polysaccharides also can be there in the fungal cell wall. But the main material, cell wall material in fungi is chitin or chitin. And there are other polysaccharides that also may be present here. Cell wall is definitely present and it is made up of chitin or chitin. That is present, and inside that you have the normal plasma membrane. Okay, nucleus will be there, all the membrane bound cell organelles are present. Okay, they don't have chloroplast, achlorophyllous, no chlorophyll, no chloroplast. Achlorophyllous, A means lack of, no chlorophyll. They don't have any chlorophyll, so they cannot perform photosynthesis. They lack that chlorophyll pigment, chloroplast also, so they cannot undergo the photosynthesis process. The next one. So they are all are heterotrophs. All are heterotrophs. Okay, they depend on others for their food. And these heterotrophs, three types are there. Saprophytes, which feed on dead and decaying organic matter, they are called as saprophytes. For this, the example is mushroom. It will be feeding on the dead and decaying organic matter from the plants, wooden logs and all. The mushrooms will grow. So that will come under an example of saprophyte. The next one, parasite. And parasites, the examples are albugo, Puccinia you got know already. So albugo, Puccinia, etc. will come here. Because they are parasitic, they will be living on the host. Host is generally plant, animal or humans. Then you have symbionts also. Symbionts means where two organisms will be living together and both of them will be getting a mutual beneficial associationship. Both of them. You give something and I will give something to you. So mutually they will be getting benefit from each other. That kind of relationship is called as symbiotism, and that organisms are called as symbionts. So they can live as symbionts also. Example for this, lichens and even mycorrhiza. About lichens or lichens, this is a symbiotic association between an alga and a fungus. Okay, a symbiotic association between an alga and a fungus, that is called as lichens. Mycorrhiza is fungus with association with roots of higher plants. So some fungi always they will be present in the roots of higher plants like gymnosperms. Okay, that kind of association where both of them will be getting benefit out of each other, that is called as mycorrhiza, symbiotic association. So here alga will be providing food to the fungus, food will be provided, and fungus in return it will be providing water, minerals, etc. to the alga. The same case even here also, fungus will be getting the shelter and food from the plant, whereas the plant is getting in return water and minerals, because they will be absorbed by the fungus and it will be giving it to the plants. This is how the mode of nutrition will be there, and they will show absorptive mode of nutrition. They are heterotrophs, so they will be showing any of these three categories, but they will show absorptive mode of nutrition. Absorption, they will be absorbing the materials. Now let us see the next point. The body is called as body of fungus. How the body of these slime molds is called as plasmodium, in the same way body of the fungus is called as mycelium. What is the body of the fungus called as? Mycelium. This mycelium will be looking like this, irregularly. Okay, this is called as a network, but this network is made up of some filaments, no? Each filament is called as hypha. So there are so many hyphae, so many filaments are there, so plural will be hyphae. So mycelium is the body of the fungus which is made up of network of hyphae. Network of filaments will be there, and that filaments are called as hyphae. So network of hyphae is the mycelium, that is called as the body of the fungus. Okay, for all the filamentous forms it will be there in the mycelium form. Then as they are multicellular, you know that they are all multicellular, so many cells will be there, filamentous forms. But in each cell, okay, this cross wall is called as septum, plural septa. This is one cell, 3 cells are there in this. So the 3 cell structure is separated by the cell wall. That cell wall only, cross walls are called as septa. So if this septa are there, then it will be showing the uninucleate condition. Uninucleate, one nucleus is there. But if the septum is not there, cross walls are not there, they are not separating the cells here, then it will be having one cell only will be having many nuclei. Here you can see 4 nuclei are there. So this condition is called multinucleate condition or xenocytic condition. It is also called as xenocytic condition. So that kind of xenocytic or multinucleate condition will come when septa are absent, the cross walls are absent. If cross walls are there, you cannot see that condition generally. Okay. If septa are present, then it is called septate, septate mycelium. If septa are not there, they are absent, then it is called as aseptate. The fungal mycelium with septa, it is called septate mycelium. The fungal mycelium without septa, it is called as aseptate mycelium. Okay, that's how it will be divided based on the nature of the mycelium. They may have septate mycelium or they may have the aseptate mycelium. Generally, aseptate mycelium will be cenocytic because there are no cross walls here. So, they will be showing multinucleate condition. Many nuclei will be there. That multinucleate condition is also called as cenocytic condition. Okay, I hope all of you understood up to here. Understood? Mycorrhiza, nitrogen fixation, no. They are not concerned with nitrogen fixation. Phosphorus absorption they are concerned with, Sanjana. Yeah, all subject corners. Cenocytic is, yeah, multinucleate condition. Tikshana, yeast is unicellular, yes. Why it is not placed in prokaryotes? Huh? It is a fungus, it shows fungal characters. Unicellular, why it is not placed in protozoa? Protozoa, they will be showing the holozoic mode of nutrition, but yeast is not showing that. It is showing absorptive mode. Holozoic means like animals, ingestion. Any further divisions in fungi? Yes, they are there. Pujita, classification is there. Which place they grow? Yeah, they grow generally in the humid, warm places. I haven't finished everything. If you understood up to here, I will continue. Yeah, they prefer to grow in the warm, humid places. Many are there in the further classification divisions in fungi. Not many, only 4 are there. What about sporangium? Yeah, I will tell. First, have you all understood up to here? Understood? Shall I continue? Do yeast have septa? No, no septa because it is one cell, unicellular. Yeah, Venkata Parao, yes. What is your doubt? Does yeast have septa? No septa for yeast because it is unicellular. Yeast, no septa because yeast will be looking like this. You must have got already the yeast diagram like this. It will show budding, small buds, yeast, no septa. Because this is the daughter, this is the parent. Okay? Understood. About morals and truffles, yeah, Venkataparao, I am just finishing the general characters. I will tell you that also, they are edible ones. But they are multicellular. I said yeast is unicellular edible fungus. If you take the mushroom, that is also morels, truffles, mushrooms, they are all edible, but they are all multicellular. Yeah, that you will get in the classification. Mycelium present in only, yes, multicellular fungi, yes. In yeast, it is not there. In rare conditions, if so many yeast will accumulate, then it will form. That is not there in your syllabus. So, filamentous forms will show the mycelium. Multicellular fungi will show the mycelium. Okay? Yes. Is mycelium present in multicellular fungi? Yes, yes. Multicellular fungi, they will be present in the, yeah, mycelium form, filamentous forms, yeah. Okay, done? Agaricus, yeah, multicellular edible fungus. Okay, understood up to here? Agaricus, mushroom scientific name. Multicellular edible fungus. That also shows that mycelium. But you have to see that mycelium under the microscope. Under the microscope. Mycelium will be there. Okay? Yeah. Agaricus bisporus. Very good, Samit. Let us see the next characters. Now, about reproduction. We have completed with the nutrition. Now, let us go with the reproduction. Reproduction in fungi, it is of mainly 3 types. Vegetative reproduction, okay, asexual reproduction and sexual reproduction. 3 types of reproduction is there: vegetative, asexual and sexual reproduction. In the vegetative, you have the types like fragmentation, Fragmentation means that mycelia will break into fragments and each fragment will become the fungus. Okay. Next, fission. Fission. In bacteria, you have learned that binary fission, one bacterium will become two bacteria. So here one fungus filament will become two fungal filaments. So that is fission, dividing into two. The next one, budding. So these all three will come under vegetative reproduction. Fragmentation, fission and budding. You know, budding is seen very clearly in the yeast. Just now before I have shown you, yeast cells will be showing the bud, external bud. This is the parent cell, parent yeast. So bud, a small daughter cell will be formed there. That is called, a process of formation of buds is called budding. So yeast is the example. So fungi will show vegetative reproduction by three means: fragmentation, fission and budding. Next, asexual reproduction. It is done by spores. Different types of asexual spores will be there, like sporangiospores. Conidia. Okay, chlamydospores. Different names are there for them, but yeah, these are the some of them. So asexual spores generally they are formed during the favorable conditions. This is performed by the fungi as they are lower organisms. This is performed in favorable conditions. So they go for in the favorable conditions by asexual reproduction, they will be reproducing by forming the spores. Remember, all these spores are haploid. All the spores, asexual spores are all haploid. Next one, sexual reproduction. Sexual reproduction also occurs by sexual spores. Okay, but before sexual spores understanding, you should know the mechanism of sexual reproduction. It occurs by the fusion of two cells or two gametes. Okay, so let us see how the fusion of two cells or gametes will be happening in the sexual reproduction. Later it will form the sexual spores. Okay, is that clear? Understood? Yes, ma? Sanjana? Yes. Okay. In vegetative reproduction, how many methods are there and what are they? In vegetative. In vegetative reproduction, how many methods are there and what are they? Three are there. Very good. Fictina, fragmentation. Very good. Fission, right? Budding. Very good. Kiran, yes. Very good, Amma. Correct. Asexual reproduction occurs by forming which structures? Yes, correct. All of you are correct. Yes. Asexual reproduction occurs by which method? Conidia, yes. Spores, basically, yes. Samreen, yes. Spores. Different type of asexual spores will be there. Chlamydospores, yes. Correct. Yes. Sporangiospores, chlamydospores, conidia. Okay, these are all different names. Very good. Now let us see sexual reproduction. Sexual reproduction involves three steps. That gametic fusion of gametes, okay, gametic fusion or cell fusion. It involves 3 steps. The first one is called plasmogamy. Plasmogamy means fusion of protoplasm of 2 cells or 2 gametes. Protoplasm means cytoplasm and nucleus. So plasmogamy is nothing but fusion of protoplasms. Of 2 cells or 2 gametes, anything you can take. So fusion of protoplasms between 2 cells is called as plasmogamy. That is the first step in sexual reproduction. Then second step is karyogamy. Karyogamy. Karyo, already you have got the meaning of karyo, nucleus. So this is fusion of nuclei. Fusion of 2 nuclei. You know, when 2 cells are fused or 2 gametes are fused, that process is generally called as fertilization, and it results in the formation of a particular structure called as zygote. When 2 gametes are fused, then it results in the formation of zygote. So that zygote will undergo meiosis in the third step, zygotic meiosis. Zygote is formed by the end of the karyogamy only. That zygote will undergo meiosis, and you know, in meiosis 4 daughter cells are formed. It results in the formation of, it forms the spores. Okay, zygote will undergo meiosis and it forms the spores. Have all understood? 3 steps are there: plasmogamy, fusion of protoplasms between 2 cells or 2 gametes; karyogamy, fusion of 2 nuclei of 2 cells; zygote will be formed, that in the third step it will undergo meiosis to form the spores. These are the 3 steps in the sexual reproduction. Okay, understood? Very good. Haploid spores, haploid spores will be formed. If zygote undergoes meiosis, then after the meiosis, the spores are all haploid. Okay. Okay, now let us see if any deviations are there in this. For example, plasmogamy should be followed by karyogamy and karyogamy should be followed by zygotic meiosis. But in some fungi, like you know, mushrooms, agaricus. I am taking agaricus as the example. In mushrooms, plasmogamy is not immediately followed by karyogamy. Not followed by karyogamy. So what happens here? When 2 cells will come and combine, okay, it results in the formation of a cell with 2 nuclei like this. When karyogamy is happening, these 2 nuclei will fuse to form the 1 nucleus. But if plasmogamy is not followed immediately by the karyogamy, then it results in the 2 nuclei, binucleate condition. This is called as binucleate condition or dikaryon stage. Di means 2 nuclei. Because in these fungi, plasmogamy is not immediately followed by karyogamy. So before zygote formation only, it results in the formation of 2 separate nuclei. They are not fused. So this one stage will be there for some time. So in the life cycle of mushroom, you can see the dikaryon stage or phase. It is also called phase. So from this dikaryon stage, the mycelium will be formed. So all the cells which are formed from this, they will have 2 nuclei or dikaryon stage will be there. That mycelium is then called as dikaryotic mycelium. From this one cell, total filaments will be formed with 2 nuclei. So that mycelium, this will form the dikaryotic mycelium, forms dikaryotic mycelium. Okay. And from that dikaryotic mycelium, so imagine this is the dikaryotic mycelium like this. All the cells are having 2 nuclei, this condition, enlarged view. Each cell in this, it is looking like this. Now this one will form the fruiting bodies. From here, the fruiting bodies will be formed like this. This total structure is now called as fruiting body. It results in the formation of fruiting body. Okay. So in the fruiting body, here that nuclei will be fused in the cells to form the zygote. So zygote is formed in the fruiting body, and zygote will undergo meiosis, reduction division in the fruiting body. It undergoes division here. Okay. First, the dikaryotic mycelium will form the fruiting bodies, and in the fruiting bodies, the zygote formation, karyogamy occurs, zygote formation occurs here. These 2 nuclei will fuse now and it will become 1 nucleus. And in this, now zygote will undergo, this is called zygote. Zygote formation also occurs in the fruiting body, and now this zygote will undergo meiosis and it forms 4 spores like this, haploid spores. Okay. But in some, zygote will become directly the spores. Okay. So speciality of sexual reproduction in the fungi, they will show the sexual spores also. They will show sexual spores. So here, whatever the spores that are formed from the zygote after the meiosis, these are all haploid spores. Okay. But in sometimes the spore will be formed directly from the zygote without meiosis. So whatever the sexual spores are there, the sexual spores which are present in this fungi are ascospores and basidospores. Basidospores. Okay, and even oospores are also there. Only oospores are 2n because zygote will directly develop into no meiosis, just directly develops into a oospore. But these are all haploid structures. Ascospores and basidospores are haploid. So sexual spores are also there, asexual spores are also there. Among the sexual spores, oospores are diploid ones because zygote will develop a thick wall around it. Okay, zygospore, other names are also there, like zygospore is there. Okay, these 2 are diploid ones. Ascospores and basidospores are formed after the meiosis. So these 2 are haploid spores. But for forming this, generally they will be showing the 3 steps: plasmogamy, karyogamy and zygotic meiosis. Okay, I hope all of you understood. If the spores are directly formed from the zygote, they are spores or that spores are diploid. Yes, they are diploid. If no division is happening, they will develop a thick wall around it. Zygote will form a thick wall around it to become the zygospore. Zygospore will be having thick wall, oospore will be having thin wall. They are directly formed. Okay. Okay. Now let us see the picture, in that you will understand this better. So all of you can see clearly in this septa. Septa are nothing but cross walls, uninucleate condition, and these are called hyphae, filaments. And here you can see multinucleate condition, it is called as cenocytic hyphae. It is aseptate. The second one is aseptate, first one is septate. Okay, first one is septate and second one is aseptate. No cross walls are there. This is also called cenocytic hyphae, multinucleate condition. There are no cross walls. If you see here, the cross walls are present. These are all cross walls. Here, that type of cross walls are not there. This is the fungal hyphae. They'll form a network to become the mycelia. This is the lichens, lichens lichens, where they will be looking like this. Okay, it is an association of alga with a fungus, symbiotic relationship. Lichens will be looking like that. This is mycorrhiza. All of you can see clearly in this. Some nodule type things are there, white color things are there on the roots of higher plants. This is also symbiotic associationship between the fungus and the roots of higher plants. Particularly gymnosperms will show. Gymnosperms. Okay. You can see here in this budding in yeast. Yeast one cell unicellular. It is forming the first nuclear division, then cytoplasmic division. Nucleus is migrating to the bud and then cytoplasm is dividing and it forms the two cells. This is the parent cell, this is the daughter cell. Then fission in yeast. It is dividing into two equal cells here. Here it is unequal. Daughter cell is smaller, parent cell is larger. Here it is equal cells, binary fission. Then these are conidia formation. Conidia are the asexual spores in Penicillium. Penicillium fungus I have shown you before. So they are the conidia. This brown color ones are conidia. Conidia are asexual spores. And these are all haploid, asexual spores. Penicillium, it will show this. Next one, the spores names, zoospores. These are the motile spores, motile asexual spores. These are all asexual spores only. Asexual spores. These zoospores are having flagella, so motile spores. Sporangiospores, the black colored ones. Then you have the chlamydospores, thick walled. Resting spores will be there, they are the chlamydospores. And then you have conidia, conidiophores, just conidia will be there, they are pinched off from the tip of the hyphae. All are asexual spores. Next, these are sexual spores example, ascospores. Okay, these are formed by meiosis. So these are haploid. Okay, bestiospores. These are also haploid. Okay. Next, zygospores. These are diploid because it forms a thick resistant wall around them. Zygospores, oospores. Thin wall will be there around the zygote. So these two are generally diploid. These two are haploid because they are formed after the meiosis. Sexual spores. Next, I was talking about dikaryotic mycelium. I hope all of you can see here the presence of two nuclei here from this stage. Can you see red dot and blue dot? This is the dikaryon stage. Dikaryon stage, dikaryotic mycelium. And then it will be forming the fruiting body. Fruiting bodies are called with the name carp, bestiocarp, ascocarp, like that. In this fruiting body, zygote is formed and zygote is undergoing meiosis to form the bestiospores or ascospores. This is how dikaryon stage will be there in the mushrooms, okay, and even in the Penicillium, Ascomystis class. So that will be there. Okay. I hope all of you understood. Done? Yes, any doubts, ma? Any doubts? Okay. Read this fungi. Yes, if you understood everything, you can give a thumbs up. Let us meet tomorrow to see the next part. What is that? Can't see clearly. Neurospores. No. That much you don't have. Neurospores and all. No. Okay. I hope all of you understood, right? Neurospora. Yes, yes. Neurospora also belongs to the Ascomystis class only. Correct. You will be learning about the classification and all later because fungi are divided into 4 classes based on the nature of the mycelium, based on the spore formation. Okay. Is that clear? Understood? Okay. Thank you, Ma. Thank you. See you tomorrow.
Hello. Good morning, students. How are you? Doing good? Everything was fine with you? Yeah, I'm fine, thank you. Okay, that's okay. Thank you for your response. That's okay. So we again meeting here in order to discuss fundamental concepts of biology, exclusively zoology. So when we met last time, we might have discussed about animal physiology, and even today also we will be discussing, we will be knowing few more points about our own human body. Done? So among these units and systems of your body, the one system which we missed last time, that was the most complex network in body, one of the most. This is body fluids, what you possess, and how these fluids get circulated in your entire body. Body fluids and circulation. The point was so simple. When we eat, food molecules from your mouth, intestine and stomach must be supplied to various body parts and organs, isn't it? Same time, when you breathe, oxygen must also be supplied to all your body parts and organs. So, both food molecules and oxygen must be supplied to each and every body part, each and every body organ, because there those organs will utilize food molecules using oxygen. In the presence of oxygen, organs, tissues, cells and perform aerobic respiration, manufacture more ATP, and they will live. But who will supply these food molecules and who will supply this oxygen to your organs? There must be some kind of tissue, some kind of fluid which is helping, which is like a river flowing from one point to another point, which is like a red river in your body, which supplies food and oxygen to all your body parts. And this main circulating fluid was the most important fluid which is supplying food and oxygen from your intestine and lungs to other body part was blood. Yes. The thing was, does blood only supply? Does it get it back also? Yes. Yes, blood will supply food and oxygen to organs, and these organs will remove carbon dioxide, will get rid of ammonia or urea, which will also be cleaned by blood again. So rather than using the term supply, we can use the term exchange, or we can use the term circulate. So blood circulates in your body, it supplies food and oxygen to organs, and it collects waste substances like carbon dioxide and urea or ammonia from your body to kidneys. So there will be a continuous network of blood and blood vessels happening in your body, which was aimed to give food, oxygen to organs and also clean these organs out from carbon dioxide and nitrogenous wastes. The main tissue, the main fluid connective tissue, which is destined to supply and clean your organs, was blood. And the interesting aspect was that blood is a kind of connective tissue, and blood tissue have matrix which is not solid. Guys, if you speak about bones are also connective, and this bone has got very solid matrix made up of calcium, phosphate, like that. And cartilage has matrix made up of sugar substances. But in case of blood, matrix itself is very fluid in nature. Therefore, the texture of this entire tissue will also be fluid. Despite being fluid in nature also, blood has got the capacity to reach every corner of your body. Yes. Therefore, blood was considered as fluid connective tissue. So, blood is fluid connective tissue. Accepted. Now, there was a question again. Is there only one type of fluid connective tissue in your body? No. Blood is like big brother. Blood has a little sister also. Yes, blood is brother, big one. There was a little sister to blood who is always helping blood. This little sister of blood, which is not red in color, which may not have RBC, but this special fluid tissue can actually provide you with immunity. Little sister of blood, which is giving you immunity, protection from various pathogens, which was whitish or colorless in appearance because it does not have RBC. What was this tissue? Lymph. So, brother and sister, blood and lymph, we can combinedly call these both substances as fluid connective tissue. Correct. So, both blood and lymph were combinedly referred as fluid connective tissue. Now, does blood flow automatically? The question comes now, does blood flow automatically? No. Does lymph flow automatically? No. Is there any organ which is controlling it? Yes. Is there any organ which is pumping it? Yes. Is there any organ which is pushing it? Yes. Is there any organ which collects it back? Yes. And all these tissues and organs were made from mesoderm, and the main organ which is supplying blood and lymph to your body parts was heart, and this heart was connected to two types of blood vessels, arteries and veins. What is a vein? Any blood vessel which pumps blood towards heart is vein. Any blood vessel which takes away blood away from heart, artery. A blood vessel which pushes blood towards heart, a blood vessel which is pumping blood towards heart was vein.
Same time, blood vessel which carries blood away from heart. Artery. So destination for vein was heart. To address for a vein was heart. From address for artery was heart. So destination of a vein is heart. Destination of artery is any organ except heart. So veins carry blood towards heart and arteries carry blood away from heart. Like this. Guys, we need to brush up, we need to learn two things here. Our prime focus must be placed on blood and its pumping organ. So blood and pumping organ. So does this heart, which is made from mesoderm, is exactly same in all animals? No. Heart is dorsal in case of invertebrates. Heart is ventral in case of vertebrates, chordates. Our heart is ventral. Cockroach have dorsal heart. Humans have ventral heart. Does every heart possess same number of chambers? No, possibly. Not possibly. If it is a vertebrate like fish, fish heart consists of only two chambers. They have one atrium and one ventricle. That was a two-chambered heart in fishes. In case of amphibians and reptiles, except crocodiles. In case of amphibians and reptiles, heart has more than two chambers but less than three chambers. Heart consists of three chambers here. They got two atriums and one common ventricle. They have got left atrium, right atrium and very common ventricle. Three chambered in case of amphibians and reptiles except crocodiles. Bit less evolved. But in case of apes, mammals, crocodiles, alligators, telling again, apes, birds, mammals, crocodiles and alligators, their heart consists of four chambers. Left atrium, left ventricle, right atrium, right ventricle. That is a four-chambered heart in case of apes, mammals, crocodile, as well as alligator. Four-chambered heart. Then, in case of fishes, guys, see this. In case of fishes, deoxy blood from body comes into atrium. This deoxy blood goes into ventricle, and from this ventricle, deoxy blood does reach gills, and gills, what will they do? Gills will give out carbon dioxide and take in oxygen. So the exchange of gases happens at the level of gills in fishes. Now this oxy blood will go to organs, and at that level of organs again oxygen will be taken up by organs, giving out carbon dioxide. Therefore, deoxy blood again goes back to atrium. So, heart of fishes will only consist of deoxy blood. Heart of fish never receives oxy blood. See, atrium to ventricle, ventricle to gills, gills to organs, organs to atrium again. Heart to gills, gills to organs, organs to heart. You can see, guys, each drop of blood passes through heart only once in cycle, isn't it? Each drop of blood passes through heart only once in this cycle. Therefore, fish heart was supposed to exhibit single circulation. That is in single circulation. In case of mammals, aves and crocodiles and alligators, left atrium, left ventricle, right atrium, right ventricle. Now, in case of aves, mammals, crocodiles and alligators, what happens was that deoxy blood from organs flows into right atrium. From right atrium, deoxy blood reaches right ventricle. From right ventricle, deoxy blood goes to lungs. At the level of lungs, oxygen will be exchanged with carbon dioxide. Therefore, oxy blood from lungs reaches left atrium. Oxy blood from left atrium will go into left ventricle, and oxy blood from left ventricle again reaches organs. Guys, see this carefully again. Always right side of your heart consists of deoxy blood. Right side of heart consists of deoxy blood. Always left side of your heart consists of oxy blood. Left oxy. So left side of heart oxy blood, right side of heart deoxy blood. Left side of heart oxy blood, right side of heart deoxy blood. Left oxy, right deoxy. Left oxy, right deoxy. Now, first blood flows through right side, reaches lungs. We call this as pulmonary circulation. Next, blood from lungs goes into left side and finally reaches organs. So first blood travels through right side of heart, then blood travels through left side of heart, which means each drop of blood is passing through heart how many times? Twice. Once through right chambers, once through left chambers. Therefore, in mammals, aves, crocodiles and alligators, of course, the circulation was double circulation because each drop of blood is passing through heart twice. Right, left. When it is going through right, deoxy blood. When it is going through left, oxy blood. Reason. Why does mammals exhibit, or what do you mean by double circulation? Definition. Each drop of blood passes twice through heart in one cardiac cycle. In one heart pumping mechanism, each drop of blood passes twice. Therefore, right deoxy, left oxy. Circulation which involves deoxy blood till lungs, pulmonary circulation. Circulation of oxy blood from lungs to left side and organs is systemic circulation. Double circulation means pulmonary circulation plus systemic circulation. Pulmonary circulation is about deoxy blood. Systemic circulation is about oxy blood. Done. This is which is happening in our body. Now, if you just go deep into details, guys, there were many concepts in this chapter. We need to learn about blood. We need to learn about heart. We need to learn about functioning of heart. How does heart contract and relax, pumping blood out of heart and receive also. And when heart is contracting and relaxing, definitely we can listen heart sounds, lub dub sound, right? Okay, and there is a mechanism or an instrument or a machine which can measure heartbeat, which can measure how much time is taken, how much time it is consuming for contraction and relaxation also. So the field of medical sciences which deals with study of heart structure, heart functioning and heart diseases was, what do you call this field of science? Field of medical sciences which deals with study of heart, heart diseases, heart ailments. What do you call this study? Cardiology. Very good. Which device, which instrument measures, listen carefully, which instrument measures blood pressure of your body? Sphygmomanometer. Simply, manometer is also accepted. Sphygmomanometer is for measuring blood pressure, correct? Now listen carefully, tricky one. Which instrument or machine or device was used to listen heartbeat? What was that? Which device or instrument was used to listen to heartbeat? Listening heartbeat. What was that? Stethoscope. Stethoscope. Yes. Stethoscope. Seth we call that as. And which device, machine was used to measure cardiac activity? ECG. Electrocardiogram, ECG. Listening is through stethoscope. Measuring is through ECG. Those are two different things. Okay, all these are done. So there are many concepts in this chapter. I will let you know briefly. So if we consider structure of heart, I will just give you a cartoon-like picture of heart only. Let us not go deep into a structure. Let us not become artists as of now. Just learn fundamental aspects because that was most important for learning things. We are not a Picasso or M.F. Hussain to draw a very good structure of heart and let you know what is it. No, that's not how we learn things. Please learn things at their base level and then try to complicate them. Always learn simplest version of a topic. If you understand simplest picture, then if you understand skeleton of the picture, then you can actually build on it. Okay, I will give you a skeleton. I will give you the fundamental diagram of a heart. which is very easy. This picture is only for understanding purpose. This picture is only for knowing what is it. I'm not going to give you a very detailed picture of heart. No, not now. We have still time for doing that. Okay. And don't please ask me, heart is red in colors, please draw in red in color. No. That's Sarvish, right? Heart was red in color, yes, of course, but that doesn't mean we should draw in red. So, heart. The anterior two chambers of heart were referred as atrium. Each chamber is atrium. So, atrium is singular, atria is plural. So, left atrium, right atrium. Between atriums, atria, there is a wall or a septum. This wall or septum separates atrium. We call that as interatrial septum or interauricular septum. In some textbooks, instead of atrium, they may use term auricles, which is synonymous but not exactly same. Atrium or auricles you can use synonymously, but not exactly. So, between atriums, we got interatrial septum, interatrial wall separates. Therefore, oxyblood does not get mixed with deoxyblood, no chance. And just drop it down. Below right atrium you can see right ventricle. Below left ventricle you can see left ventricle. So, right ventricle, left ventricle. Guys, you can see wall of a ventricle is comparatively much thicker than wall of atriums. And thickest wall is with left ventricles. This is just a cartoon-like depiction of a heart structure, not exact picture. First learn basic things, that's it. Okay, simple. Let it remain as such. And you can see here, there was again a septum between both ventricles also, right? What should you call the septum? This is interventricular septum, right? Interatrial septum, interventricular septum. These septums does not allow mixing of oxyblood with deoxyblood. These septums does not allow mixing of oxyblood with deoxyblood, but between right atrium and right ventricle there is a septum which is called as right atrioventricular septum. Right atrioventricular septum. Same time, between left atrium and left ventricle also, there was a septum here which was referred as left atrioventricular septum. So, right atrioventricular septum, left atrioventricular septum. These atrioventricular septums are not completely closed, they are open. See? So, oxyblood flows from left atrium to left ventricle through hole in septum. Same time, deoxyblood is also flowing from right atrium to right ventricle through this hole in septum. But these holes, openings, are not always open. They are like gates. They may open, they may close. Guys, these are permanently closed, but these are temporarily open, temporarily closed. But how? Right septum, right side, there was a valve which is guarded by three flap-like tissues like this. Zoom it, I'll show you. It's a valve which is guarded by three types of flaps like this. Three muscular elastic flaps. So as there are three cusps, three muscular elastic structures. So this right valve is called as tricuspid valve. Similarly, on left side, this opening was protected by a valve which is consisting of two cusps only. Two cusps. Therefore, this is bicuspid valve. And it has got also one more name. If you see, there used to be hat, cap of some English persons whose cap was like this almost. Cowboy caps, like this. So, their cap from top you can see structure like this. Their caps have this depression. This is called as mitrum. As this valve also looks like the cap, this bicuspid valve was also referred as mitral valve. Mitrum. Okay. So you got chambers. Left, left, right, right, deoxyblood. Deoxyblood in right side of your heart, oxyblood in left side of your heart. Accepted. Done? Now, so veins, inferior vena cava and superior vena cava, superior vena cava veins, inferior vena cava veins, they will supply deoxyblood from body parts, superior from upper body parts, inferior from lower body parts. They will pump deoxyblood into right atrium. From right atrium, deoxyblood goes into right ventricle. And right ventricle will have a blood vessel. Right ventricle supplies deoxyblood to lungs. What do you call this? Pulmonary aorta. Done? Pulmonary aorta. And lungs will exchange carbon dioxide with oxygen. And oxyblood from lungs comes into left atrium through pulmonary veins. Okay. and oxy blood from left atrium goes into left ventricle, and from left ventricle oxy blood goes into different body parts through systemic aorta. Like this, it will bend. Okay. So pulmonary vein comes from right ventricle, systemic vein or systemic aorta comes from left. This is systemic aorta. This is pulmonary aorta, this is systemic aorta, like this, one above the other. Okay, like this. Done. So just stick to basic diagram, basic explanation. Once you understand these fundamental aspects of heart and their different parts of it, which is receiving oxy blood, which is pumping oxy blood, like this. Same time, at the base of pulmonary aorta and systemic aorta also, there are half moon shaped valves. Half moon, half, semi, moon, lunar. What do you call them? Semi-lunar valves. Guys, when tricuspid and bicuspid valves close, you get first heart sound lub. When semi-lunar valves close, you get second heart sound dub. That's it. Stick to this. Lub sound is produced due to closure of tricuspid bicuspid valves. Dub sound is produced or listened when there is closure of semi-lunar valves. Okay? Let us conclude at this point. You need to learn basic aspects. Understood? Chambers, vessels, valves and sounds. Okay? Yeah, both are by ventricles. Yes, Raman, Pole. Lub and dub sound both are made by ventricle. Yes, correct. Except, yes. Good. Okay? So we'll conclude today and we'll learn fundamental aspects about excretion or kidneys tomorrow. Okay? Thank you.